All right, let's get started here. So welcome everybody to the AP Research Roundtable. This is our October roundtable, and we're really excited to have today's speaker, who's actually also a researcher, part of the core group of researchers that have come together over the last year and are really driving forward some of the major tracks of our segments, I should say, segments of our research, AP research strategy. So Professor Hiruy Meharena from UCSD, I'm really excited to hear what you have to say and would love for you to introduce yourself maybe a little bit. I know you're a regular attendee here, which is something we really appreciate from our AP researchers, that they not only present, but also are attending and are engaging on both sides of the presentation. So maybe a little bit about, again, about your where you are and how you kind of came into AP, maybe a little bit about your background and in CNVs and what makes you such a special member of this community. So please, without further ado, let's jump right into your conversation, and then I will reserve some updates for the very end in terms of Project AP and some research sort of strategy updates, but we'll save that for the very end. So with that, please, Rory, take it away.
Thank you for the wonderful introduction, Ethan. And can you all see my screen?
Yes.
Great. So as Ethan mentioned, I'm one of the faculty working on, as part of this larger cohort of AP groups. My lab is located at UCSD. We're in San Diego, and a few other labs are also here. So we work collaboratively together. My research endeavor into AP came through my work with Down syndrome. So we're really interested in understanding the converging features of genomic imbalance disorders in neurodevelopmental intellectual disabilities. And today I want to share with you some of the work that we've done in the past. And this is mostly coming from the work I did as a postdoctoral fellow when I was at MIT. And I'll also give you some insight into what my lab is working on now. So just to give a brief overview, the brain is composed of multiple brain cell types. It's one of the most complex organs in the human body. And as you can see here, we have our neurons, which is what we typically seem to think about when we think about the brain. These are the functional cells of the brain. But there are other cell types as well. These are collectively known as glial cells. There are three types. There's oligodendrocytes. These are the ones that insulate the neurons so that there's no short circuits that are occurring in the brain. And when two neurons communicate, the communication is pointed in a very specific manner. And then astrocytes are the support cells of the brain. They provide nourishment. They provide neurotrophic factors that allow these different cell types to function properly. They also are the communication between the blood system or the vasculature system and providing food and nurture to the rest of the brain. And microglia, which originate from a very different lineage of the cells that are during development. And these are the immune cells of the brain, ensuring that there is no debris or cell death and just regulating or infection in the brain. So collectively, this is what makes our brain. So my lab is really interested in understanding intellectual disabilities, and predominantly we're interested in genomic imbalance intellectual disabilities. So this includes aneuploidies. Most of you probably have heard of Down syndrome, which is caused by the triplication of chromosome 21. We have Edwards syndrome and Patau syndrome that are triplications of chromosome 18 and 13. These individuals basically have an extra chromosome of the specified number. But we're also interested in copy number variations, which includes AP syndrome. But there are approximately 11% of individuals with autism have some sort of copy number variation. This includes either a deletion of a specific region where, for example, here, this yellow is part of the original and it's lost in individuals and you can see it's much shorter, or you have an insertion which is the opposite direction. And collectively, copy number variations account for the largest group in autism spectrum disorder. And nearly all individuals with copy number variation have some level of learning disabilities. So today I want to focus on our work that was looking at trisomy 21, and we looked at the 3D genome architecture, and I'll give you an introduction of what that would look like, and then how it impacts neuroprogenitor cells, as these have been implicated as the pivotal cell types in development that are highly impacted by aneuploidies or genomic imbalance. And then I'll cover some of the ongoing projects that are in the lab, and then how that all of this work that we're doing fits in with Project AP and how we're aligned. So in Down syndrome, studies looking at postmortem human brain samples have identified that there's a subset of regions that have significant hypocellularity. And hypocellularity means that there's a loss in the number of cells within those regions. And what studies have found is that the frontal and temporal lobe, which is this cortical region, which we utilize for processing information, thinking, making executive decisions, seems to be hypocellular. The hippocampus, which is where the memory is significantly processed and is believed to be a pivotal part of the brain that is involved in memory and learning. And then the cerebellum, which is involved in numerous other features as well. And so these are 3 brain regions, at least in Down syndrome, that have been associated with or have been shown to be implicated in the learning disabilities in individuals with Down syndrome. And at the cellular level, studies have shown that there's significant reduction of proliferation. This is just the multiplication of these neuroprogenitor cells. Neuroprogenitor cells are the cells that give rise to neurons, astrocytes, and oligodendrocytes. And it seems that this hypocellularity is associated with reduced proliferation during the mid-gestation period during development. Another feature that has also been observed at the cellular level is that individuals with Down syndrome have a higher number of astrocytes, these are glial cells, as compared to the neurons. So for some reason, triplication of chromosome 21 seems to shift the differentiation towards an astrocytic lineage, resulting in reduced neurons. So, and this imbalance of the different cell types has been associated with learning and memory disabilities as well. The third one, which has been predominantly looked at utilizing a mouse model for Down syndrome, which is called TS65DN, and other mouse models as well, is that there's significant reduction in the migration of these neuroprogenitor cells during development. And we require cell migration of these neuroprogenitor cells because we need a specifically layered cortex that allows us to have a good connection with other regions of the brain. And in addition to that, we also observe that this mouse model has a smaller brain as compared to the neurotypical or the mouse that doesn't have aneuploidy, indicating to us that there's a good combination of mouse models and postmortem human cells or tissue samples that have been utilized to get a good understanding of the cellular level. However, when we think of what are the molecular mechanisms that are governing all of these cellular and organ level manifestations, things have been very difficult. We have not been able to identify specific genes on chromosome 21 that are driving these features. And with the advancement of next generation technology, studies have started finding that, in fact, triplication of chromosome 21, the alterations in gene expression is not limited to the genes on that chromosome, but are actually spread out. So when we compare samples that were derived from individuals with Down syndrome and age-matched neurotypical individuals, we see that there's significant changes that are occurring genome-wide. And in addition to this upregulation of those triplicated genes that are expressed in that cell type. So when I first started my postdoc, I was really interested in understanding how the triplication of chromosome 21 impacts the nuclear architecture. So if we think of the nucleus as this elevator and every chromosome as these individuals in the elevator, then my thought was maybe the triplication of chromosome 21 would require every other chromosome inside that elevator to reorganize themselves in order to accommodate this third chromosome. And that might be associated with the genome-wide disruption in gene expression. So just to give you a brief introduction of how the genome is organized inside the cell. We have 23 pairs of chromosomes, and this is about 6 feet of DNA. And this 6 feet of DNA is wrapped around histones, allowing it to condense. And this 6 feet of chromatin, which is this DNA histone complex, needs to fit into this very tiny structure, which is called the nucleus. However, in the past, we thought that the organization of the DNA inside the nucleus was very random. But with the advancement of microscopy and next generation sequencing approaches, we've now identified that every chromosome actually occupies a very specific space within the nucleus during interphase. And this organization of the nucleus, or the genome inside the nucleus, is known as chromosome territories. And one of the interesting things about chromosome territories is that every single cell type has its own specific chromosome territory occupation. So if we think of neurons versus astrocytes or skin cells or muscle cells, each one has a very specific organization of where the chromosomes are located within that spatial context, indicating to us that the localization of the chromosome itself plays an important role in defining the cell type identity. And so if we were to cut this chromosome in half and flip it 90 degrees, what we would see is that there are two types of interactions that occur between these chromosome territories. So if you think of every color as representing one chromosome, there are cis interactions. These are the interactions that are occurring within the chromosome itself. And then there are trans interactions. This is the interaction that happens between two chromosomes or neighboring chromosomes. And if we took a deeper look into a very specific region, there are these structural units of the genome known as topologically associating domains. So these are structural units that every cell has, and by reorganizing where these topologically associating domains are, we can change the gene expression levels. For example, towards the nuclear periphery, it's known as the inactive compartment or B compartment. This is where the topologically associating domains that have genes that are not required for that cell will be localized. And towards the nuclear core, this is the topologically associating domains that harbor the genes that are required for that cell type will be localized in the active or A compartment, and these genes will be expressed, whereas the ones in the B compartment will be silent. One of the interesting things that happens during differentiation or development is as we go from, let's say, a pluripotent stem cell state, this is the early cells that are found in embryogenesis. If we were to, for example, to have two topologically associating domains in the B compartment and two topologically associating domains in the A compartment, what we find is that as we transition from the pluripotent to the progenitor state, we observe some compartment switching, and this is not complete compartment switches, where we observe topologically associating domains switching from the B towards the A and 3 going in the opposite direction. However, this is still an unstable sort of positioning where they're both occupying both spaces at the same time. And once we get to a fully differentiated cell, which would include cells such as neurons, astrocytes, and the other cell types of the brain, these topologically associated domains are now stabilized in where they're supposed to be. And studies, like I mentioned, have indicated that the neuroprogenitor cells in individuals with Down syndrome are the most impacted by that triplication. So we wanted to understand, what is the consequence of triplication of chromosome 21 on the 3D genome architecture as we transition from the pluripotent stem cell to the progenitor state. So just to give you a brief introduction of how we look at the 3D genome architecture. So as you can see here, and as I mentioned, there are cis, these are interactions that occur within, and there are trans, which are interactions that are occurring in between. So for example, if we had an interaction between the purple chromosome and the red chromosome here, the ones that are in close proximity, we can have them bind together, and then we can shear the DNA into small pieces. And then we circularize this DNA, and then we cut it apart so that there is this one chimeric molecule that has now a region from chromosome purple and another one from chromosome red. And then we can utilize pair-end sequencing to actually see where these interactions are coming from. And so now we can lay out every single chromosome. Like I mentioned, we have 23 pairs of chromosomes. So if we have them on the horizontal and vertical in the same way, we observe that there's significant interaction within a chromosome. These are the cis interactions, where every pixel represents an interaction. But we also have trans interactions. For example, here, this is a trans interaction between chromosome 1 and chromosome 3, which is shown as this highly pixelated region. So we want to understand how this 3D genome architecture is being altered by chromosome 21, triplication of chromosome 21. So we utilized human-derived induced pluripotent stem cells to generate both stem cells, and then we differentiated these stem cells into neuroprogenitor cells. Here you see that we've stained them with nestin SOX1 and PAX6. These are hallmark markers for neuroprogenitor cells, as well as OCT4 nanog for induced pluripotent stem cells. And the top panel represents euploid cells. This is genotyping or karyotyping, where we observe that there are only 2 chromosome 21s, and the bottom panels represent the trisomy 21 derived samples, where there's 3 chromosome 21s, as you can see here. And we generated these from both an isogenic sample, where the individuals have both the euploid and the trisomic cells come from the same individual, minimizing genetic variation in our studies. But in order for us to ensure that what we are observing in this isogenic pair is also recapitulated in a larger cohort of individuals with Down syndrome, we took non-matched but just sex-matched individuals, both male and female, from euploid and trisomic individuals. So we performed our Hi-C, or this 3D genome analysis, and next I'm going to show you this differential analysis of the interaction maps. So as I mentioned, there's significant alterations as we go from induced pluripotent stem cells to neuroprogenitor cells. This is a map showing the difference in neuroprogenitor cells and induced pluripotent stem cells in the euploid individuals, where red indicates that there's a significant increase in interaction in neuroprogenitors, and blue represents a significant reduction in neuroprogenitor cells as compared to induced pluripotent stem cells. And as you can see, there's this checkerboard sort of pattern where there's a reorganization that's occurring just genome-wide. However, if we did a similar analysis, looking at the cells derived from individuals with Down syndrome, what we observe is that there's a significant loss of the trans chromosomal interactions. So there's significant reduction in interactions that are occurring in neuroprogenitor cells between the different chromosomes, and there's increased interaction with themselves. And so we wanted to understand whether this alteration is happening in the induced pluripotent stem cell or the neuroprogenitor cell state. We just performed the distribution of these cis and trans interactions. And what we observe is that in induced pluripotent stem cells, there isn't a significant alteration due to trisomy 21. However, in neuroprogenitor cells, we see significant reduction in the trans chromosomal interaction. This is the interaction that is shown here in between the chromosomes, and significant increase in the intrachromosomal interactions, which is the interaction with themselves. So if we were to animate this, for example, what we would observe is that with the triplication of chromosome 21, every chromosome becomes introverted and spends more time interacting with itself than it does with its neighboring chromosomes. So we were interested to see if any other studies have actually observed a phenomena that was similar to this. And interestingly, what we found is that in 2016 and 2018, there were a few studies that came out looking at senescent cells. And I'll explain what senescent cells are, but what they observed is that there's these shorter range interactions, less than 2 megabase in range, in senescent cells that isn't altered in proliferative or quiescent cells. These are quiescent cells, are cells that have exited cell cycle. And this is very similar to what we observe in neuroprogenitor cells, where there's significant increase in these shorter range interactions and a decrease in these longer range interactions. So are these cells becoming senescent? So the first question that we wanted to understand is, what is senescence? And senescence was first described by Leonard Hayflick. This was in the 1950s and 1960s, where they observed that there's a cell has a very limited number of times that it can divide within a dish. And this was associated with replicative exhaustion and associated with aging. And one of the key features Leonard Hayflick and colleagues identified was that cells permanently exit from cell cycle. This is reduction of proliferation, and they become anti-apoptotic. So these cells are actually do not die once exiting cell cycle. But since then, several studies have looked at various features, such as the changes in the 3D genome organization, chromatin structure, nuclear lamina, and changes in gene expression, as well as accumulation and DNA damage. In addition to that, there are organelle dysfunctions, such as mitochondrial and lysosomal dysfunctions. And these cells are actually toxic to the tissue microenvironment because they release this senescence-associated secretory phenotype. And one of the interesting things that we observed is that some of these key features have also been observed in studies of individuals with Down syndrome. As I mentioned before, reduced cell cycle or proliferation has been observed in the brain and other features, organs in individuals with Down syndrome, as well as the mouse models representing Down syndrome. Changes in nuclear architecture, as I've shown you in our neuroprogenitor cells. We've looked at chromatin states, nuclear lamina, changes in gene expression, as well as accumulation of DNA damage. And there's also significant changes in mitochondrial function. It's one of the hallmarks of individuals with Down syndrome, as well as increased DNA damage, indicating to us that there might be some similarity in terms of senescence and individual in neuroprogenitor cells that we see with triplication of chromosome 21. So even though originally senescence was associated with replicative senescence or replicative exhaustion, since then, numerous studies have shown that there are several ways that senescence may occur. And this includes oxidative stress, oncogene, which is cells that have cancer have a higher tendency of going into senescence, as well as chemotherapy, which is and radiation for cancer therapy also induces senescence. So there's been several studies that have actually performed RNA sequencing from all of these different sorts of senescence entries. And so we wanted to do a comparison of all of these previously published RNA sequencing, and we've done exhaustive RNA sequencing in both induced pluripotent stem cells and neuroprogenitor cells. And so just looking at some of the hallmark classical markers, such as P16, P15, P21, that are upregulated in senescent cells as compared to non-senescent cells. Interestingly, we observe this sort of signature only in neuroprogenitor cells. We see some senescence-associated secretory phenotypes that are upregulated in senescent cells, also becoming upregulated in our neuroprogenitor cells that harbor trisomy 21. And there's a few nuclear markers that are classically downregulated in senescent cells, and we also observe these similar signatures. So, and these features are not present in induced pluripotent stem cells, as you can see here, both from our study and another study as well. But this is just looking at a few markers. So what we've done now is we've actually expanded it to all of the gene expression changes that are occurring. So we looked at the up and down regulated genes and how much overlap there is between, the up and down regulated genes observed in induced pluripotent stem cells and also neuroprogenitor cells. And what we find is that the senescence-like signature observed in neuroprogenitor cells is very similar to the senescence features observed in oxidative stress-induced senescence, indicating to us that globally, these cells might be exhibiting some sort of oxidative stress-induced senescence phenotypes. So the first thing that we wanted to do is confirm that these neuroprogenitor cells that we have also show some senescent features. So we've performed senescence-associated beta-gal staining, and we observed that there's a significant increase in the number of neuroprogenitor cells that harbor trisomy 21 entering deep senescence, which is marked by severe upregulation of this senescence-associated beta-gal staining. And so we wanted to see if there are any therapeutic avenues that we can utilize to mitigate some of these features. And one of the most interesting things is in aging, senescence has been one of the driving features and a good target for therapeutic avenue. A lot of this work was driven by James Kirkland at the Mayo Clinic, and they were able to identify a combination of a kinase inhibitor and an antioxidant, which is dasatinib and quercetin. And if you use these two drugs in combination, they've shown that you can eliminate and also alter senescent cells. And they've done some clinical trials in individuals with diabetic kidney disease, and currently they have studies, clinical studies in both Alzheimer's disease and Parkinson's disease, which we're hoping to see the results soon. But we wanted to use this drug combination to see if we can ameliorate some of the cellular and molecular phenotypes that we observe in these neuroprogenitor cells harboring trisomy 21. So we treated our neuroprogenitor cells for about 5 days, and then we interrogated a few of the features to see if we can get rid of these senescent cells in the beginning, to look at some of the transcriptional changes as well as some cellular phenotypes. So the first thing that we wanted to do is utilize a senescence marker, which is P16E4A, is one of those markers. And what we observe is that there's a significant increase in P16 positive neuroprogenitor cells in trisomy 21 harboring neuroprogenitor cells. And after dasatinib quercetin treatment, we actually are able to rescue these to the similar levels as those observed in euploid cells. So we are able to eliminate these cells. Just to give you a brief reminder that some of these hypocellularity in the different brain regions have been associated with proliferation defects as well as migration defects. And we have some data in our ongoing studies looking at altered differentiation, but in our published data set, we interrogated some of these proliferation and reduced migration changes. And one of the most interesting things that we saw is that we performed RNA sequencing after drug treatment. And interestingly, we were able to observe that we were able to ameliorate approximately 54% of the differential gene expression that we saw. And two of the features, which include cell migration and cell cycle, were in the clusters where we were able to ameliorate it with this dasatinib quercetin drug treatment. So we wanted to see if we are able to ameliorate these changes at the cellular level. So we performed Ki67 staining. This is just looking at the number of proliferative cells at that given time point. And what we observe is that there's a significant upregulation or increase in the number of proliferative cells after T21 neuroprogenitor cells have been treated with dasatinib and quercetin. And we also performed migration assays. And as you can see here, these are the euploid cells. All of these green dots represent a cell, a neuroprogenitor cell that has migrated out of its neurosphere. And as you can see here, there's significantly reduced number of migratory cells. And after dasatinib and quercetin treatment, we're able to see that we can ameliorate at least the distance that each cell is migrating, even though the number is still slightly less than what we would expect for the euploid samples. So just to give you a summary of what I've talked about so far is that we found that trisomy 21 uniquely induces neuroprogenitor senescence, and we're able to ameliorate some of these disruptions that are introduced by trisomy 21 utilizing the senolytic drug combination of dasatinib and quercetin. So one of the questions that we had was, is this senescence phenotype actually observed in all of the brain cell types after neuroprogenitor cells? So we utilized induced pluripotent stem cells derived from individuals with Down syndrome. We've generated neuroprogenitor cells, as I've showed you so far, and we've gone further and we've generated all these different cell types. And what we found is that here is just to show you our system. We've optimized these protocols so that we get cells that actually mimic what we observe in the brain morphologically, as well as at the molecular signature level. And microglia, like I mentioned, are derived from hematopoietic progenitor cells, as shown here in our cartoon. So they have a different lineage. So we wanted to understand if this whole system of senescence is a progenitor phenotype that actually transduces to the later phenotypes as well. And so this is just looking at the RNA sequencing data sets. And what we can see is we can differentiate these different cell types based on their transcriptional signature, global transcriptional signature. So on the left, you see induced pluripotent stem cells becoming into neuroprogenitor cells that become into astrocytes. And then towards the right, we see that there's a significant alteration as we go from stem cells to hematopoietic progenitors. And on the first principal component, there's a significant similarity. So we can map these trajectories in this way. Interestingly, what we observe is that all of these different cell types have genome-wide disruption, where induced pluripotent stem cells have the lowest number of differentially expressed genes, and neuroprogenitor cells have the highest. Microglia have the second highest differentially expressed genes, and astrocytes and hematopoietic progenitors are in between induced pluripotent stem cells and the other cell types. So we wanted to take this whole RNA sequencing data sets that we've now generated for individuals with Down syndrome and assess if senescence is actually happening in all of these cell types or in a subset of these cell types. So just to give you a brief reminder, there's these classical senescence markers, which are P16, P15, and P21. There's the senescence-associated secretory phenotype signatures that are also upregulated. Red means it's just upregulated, and then these nuclear markers that are downregulated. Interestingly, what we observe is that induced pluripotent stem cells do not have this senescence signature, and neither do any of the other cell types. So it seems like neuroprogenitor cells are the ones that are uniquely expressing these senescence-like signatures. However, even though there is genome-wide disruption of gene expression, none of these other cell types seem to have the senescence-like signature, indicating to us that this genome-wide disruption in gene expression may not directly be associated with senescence itself, indicating that Trisomy 21 may utilize different pathways to induce this genome-wide changes and dysfunction in cell type specific manner. So these are some of the things that we're currently working on, trying to figure out and try to see if we can develop therapeutic avenues that would be applicable during development and during adulthood, and maybe even during aging. So this is some of the ongoing work that's in our lab. So how can this apply to our future studies utilizing the cells derived from AP heroes to get a better understanding of where AP duplication is disrupting the process of neurodevelopment, learning and memory, and which cell types are most altered by the system. And so the timeline that we're looking at is for the first year, we want to focus on induced pluripotent stem cells and neuroprogenitor cells, like I showed you in most of the studies that we've done so far, to get a better understanding of how duplication of this 20 megabase region of AP impacts the 3D genome architecture, the transcriptome, and the epigenome of the different cell types of the brain. And we're hoping that we can utilize these data sets that we generate to identify assays that we can utilize to identify novel therapeutic avenues that would be helpful for individuals with AP. And then we would like to expand this approach the way we're doing for Down syndrome and all these other genomic imbalance disorders that we're working on to try to get a better sense of how AP disrupts some of these systems and cell type functions. With that, I would like to thank my group, as well as Niwei and her lab at MIT. This is where I did my postdoc, and all of the individuals listed here have contributed significantly to the work that I presented today. And of course, I'd like to thank Bina, Ethan, and Whitney for having this great roundtable where we are able to discuss candidly about the thoughts and where we're trying to go, and also for the kind and generous support for our research endeavors. And I'd like to thank all of you for taking the time to be here today. And I'll take any questions. Thank you.
Awesome. Thank you. Thank you for that energy and clarity. So Jason, you had a question in the chat. You've been waiting patiently. Next time, I think, feel free to just chime in. I think we want to encourage polite, like a lab meeting, so we don't want to break someone's train of thought, but get in your question, when you feel like you should ask it. But let's come back to you, and then I have a question, and then let's open it up to the audience. So go for it, Jason.
Sure. So that was a really fascinating talk. Thanks so much for that. I was wondering about the chromosome, the high C experiments, whether you were able to extend them in vivo, and to what extent the OCT4 overexpression, reprogramming, in vitro culturing might be influencing the patterns you're seeing.
Yeah, that's a really interesting question. So for Down syndrome, it's pretty good because we have really good mouse models now. So we are currently doing this. But one of the interesting things is that even though we've thought of senescence as this aging feature, studies have actually found that there is this mid-gestation senescence that occurs throughout the body. So there's these senescent cells that happen, let's say in a mouse would be between, E13.5-ish, which would be that mid-gestation period, and it goes away during late gestation. So our hypothesis is that we actually think that, population of cells is expanding in Down syndrome. And that would alter, the cell type specificity and all these things. But we are utilizing the mouse model to now explore, how this transition happens during development. But we don't have any results. But that's a great question. Thank you.
So you're saying that there's a kind of program senescence? Or I'm trying to understand that. Okay, that's a little bit blowing my mind, I think.
Yeah, so we now have a mouse model where we can track senescent cells when they're born, and we want to understand what they become in the future. So this is one of the key projects that's ongoing in the lab. And what we're hypothesizing is that senescence naturally occurs during mid-gestation, and genomic imbalance actually maybe expands or holds these cells in that deep state for a longer period of time, which means that during mid-gestation is where all your neurons are born, and late gestation is where all the glial cells are born. So if you were to shift this whole system in that direction, then it would, hypothetically would explain why individuals with Down syndrome have a higher number of astrocytes and reduced number of neurons, because you're prematurely making these neuroprogenitor cells enter senescence. So that's a hypothesis we're working on. We're trying to attack it from multiple different points.
Okay, Arya, you're up.
Hi, nice talk. So can you say something about correlation versus causation in these high C patterns that you see and differences in gene expression? Like, do you think that the mispositioning of things in the nucleus is really upstream of the differences in gene expression, or maybe a consequence of some misexpress or having different dosages on on chromosome 21, and why do you think it's particularly sort of relevant to look at these high C things?
So the interesting thing is that the genes that are on chromosome 21 that are upregulated, approximately 87% of the genes are the same in induced pluripotent stem cells and neuroprogenitor cells. And that 13% are these low expressed genes, and none of them are transcription factors or any sort of, cell signaling hubs that might have that. And we're still exploring this idea. But what we do know is that the introversion happens in specific TADs, and the TADs that are becoming introverted are the TADs that are highly enriched for genes that are becoming upregulated. And the TADs that are losing, which are a very small subset of them, these, losing long-range interactions, the genes in there are enriched for downregulated genes. So we see that there is a correlation, right? And we have these DCAS systems that we built now to try to see if we can actually disrupt these long-range interactions and see similar features in that way. But in the field of schizophrenia, right, studies have shown that if you break long-range interactions, you can actually change the gene expression levels. But in our case, right, we got to the senescence point through this different route. We weren't really looking for senescence at the time. Is that the 3D genome architecture led us to this phenotype. And so, we're interested in the 3D genome just to get a better understanding if genomic imbalance could do that. And we're utilizing human artificial chromosomes to get a better understanding if it's genes or genetic material or the combination of both.
Yeah, I mean, I'm not an expert in this field, but in development, I know it's been sort of very hit or miss how important the architecture is. Like, there's lots of cases where people perturbed what people think of as really important sort of interactions where you see these very robust domains and it had no effect, right? Sometimes it has an effect, but I think, some people think that sort of most of the architecture has to do with the way, you need, as you said, you need to package this really big thing into this really small space. And so maybe it's more of a solution to that problem than anything else.
But if you think of, mutations such as CTCF mutation, cohesonopathies, laminopathies, right, where the 3D genome architecture is significantly disrupted, those individuals at broad strokes have similar features as individuals with Down syndrome, right? Craniofacial abnormalities, hypotonia, heart defects, intellectual disability, the brain development, the regions of the brain are very similar. And this is, we have those cell lines now. So we've generated CTCF mutations, lamin mutations with, those mutations associated with individuals with intellectual disabilities that are similar to the individuals with Down syndrome. So we are looking at it from both angles, but I don't think it's, one single thing. I think here is this really broad stroke changes that are occurring, whether it is this one promoter enhancer interaction or some other significant reorganization that's altering the system. We don't know.
Okay, thanks. If I could ask one more quick.
Go for it. Go for it.
It seemed to me, maybe I misunderstood, you're looking for therapeutics that would overcome this senescence phenotype. Do you worry about, like, maybe the program senescence sort of has some biological rationale, or maybe there's some damage in these cells that necessitates this phenotype? So just, having these cells that still have these genome alterations, but just overcoming the senescence, do you think there might be sort of, I don't know, side effects with that or something?
Yeah, I mean, definitely, right? Because if we push senescence too far off and we reduce it, then you'll have less glia, which is also problematic. And when you target them, you're allowing these cells to continue proliferating. So it might cause different types of cancers. So there are these issues. But beyond that, right, dasatinib, which is a kinase inhibitor, is just massive toxicity. So it's pretty hard to utilize any kinase inhibitors or therapeutic avenues in this way. So we're not saying that this is what we need to target, but we're saying that we're expanding, the cellular phenotypes that can be utilized for screening sort of purposes. And we're trying to see if the cells derived from individuals with AP show similar features or different features, right? We don't really know. That's why we're taking this very broad approach to actually characterize, the stem cells that we're getting from AP.
Thanks.
So let's get to Nicoletta 1 sec. I just wanted to maybe follow on here. So why are neuroprogenitor cells so sensitive to either trisomy 21 specifically or trisomy maybe generally? Why, like in laminopathies that have, that manifest as dilated cardiomyopathy, you can understand that the muscle progenitor cell is physically turning into a muscle fiber, and that's going to crunch the genome and but what's going on? Is there something physical, or what else is, or the trophic factors? Like, why is it about NPCs you think that sensitize them to trisomy?
Yeah, I mean, the honest answer is we have no idea. I mean, I can hypothesize a billion things, but
all right, what's your top hypothesis?
I mean, I think one of the key features is that progenitor cells are in transition, and I think when you look at hematopoietic progenitor cells, their mechanism is very different from neuroprogenitor cells. For example, when you look at the nuclear lamina, the hematopoietic progenitor cells are already utilizing lamin A, whereas neuroprogenitor cells are still utilizing lamin B. So maybe that's that. Maybe the 3D genome architecture is in transition and doesn't have a really stable state, and therefore altering it might shift things this way or that way. Or it could be where the different genes are located, right? What we find is that astrocyte-specific genes or glial-specific genes in neuroprogenitor cells are towards the nuclear periphery. So if you were to dislodge these, nuclear lamina-associated domains, then you're allowing upregulation of, astrocytic genes, therefore shifting it in that way. Currently, we have no idea.
Those seem like really solid leads, though. So I was hoping you might share. Nicoletta, thanks for being patient.
Hello, thank you very much for your fascinating talk. I just have several questions, actually, but I'll take it slowly. So I'm thinking about the process of going to these stem cells. Are you using them from fibroblasts? Are you deriving them from fibroblasts, the stem cells?
We have them from both. We have from both fibroblasts and PBMC.
And are the fibroblasts having a senescent phenotype that can be correlated in any way with this neurological senescence, neuroprogenitor senescence?
That's very fascinating. We've never looked at
and
fibroblasts.
Is there any individual variability that you observe in your differentiated cells senescent processes? If you do neuronal progenitors from 7 lines, how do they behave?
So all of them will show you senescence phenotypes. But when you look at the proliferation rate, there's a significant spectrum, right? Like they're all less than the euploid cells, but you see that the severity, it can be variable. So when the cells that we derive from individuals with mosaic Down syndrome, which are adults, their proliferation state is much higher than the cells that were derived from individuals that were just spontaneous termination. So there is where the cells come from seems to have difference in severity. Even in gene expression profiles, you see that, if you have euploid on a PCA plot, the mosaics individuals, and then, as you go further and further away, the severity seems to be further. Yeah, we don't really understand why, right? Because everybody has triplication. We technically purged the epigenetic signatures by converting them back to iPSCs. But this is something that we're exploring right now.
Thank you very much. Thanks a lot.
Thank you.
Faye, do you have any questions, or any of the families have any questions? Feel free to ask.
Thanks. Yeah, so I have lots of questions, but when they kind of like disappear from your mind, that was just a brilliant talk, and I didn't know about the organization of the 3D genome, the chromosome territories. That's all kind of news to me. You mentioned increased DNA damage in trisomy 21. So I think when I was thinking about that in the context, are there like particular hotspots that you would see that sort of like, almost add to that cross-linking experiment that you showed?
Yeah, that's a great question. And that's something that we're looking at. So far, what we do know is that the type of DNA damage is very specific. So we don't see any double-stranded DNA breaks. What we're observing is that increased oxidative stress associated lesions. So these are single-stranded DNA breaks, and we're doing single-strand DNA break mapping. So we don't know exactly, and we're trying to do this at super high resolution to get an idea if there's significant variability. So we're trying to do it at single cell level to see if each cell is breaking at the same point every single time, or there's just these random breaks that are occurring all over so that you can't really see like a specific location coming up, but that's something that we're looking into.
Great, thank you. And so, I mean, you really have blown my mind here, right? So with the chromosome territories, so if you took like, a human body and you just like plot literally cells from every different organ, would they all have that same configuration? No, each cell will have its own configuration. So you can look at each configuration and say skin, muscle, heart. So that's, this is some of the studies that looked at chromosome territories come from imaging experiments. So they were looking at whole chromosome painting, and you can see that a heart cell has this and then that. Yeah, so you can define them in that way. Okay. I probably learned it once and just forgot about it. So brilliant. Anyway, thank you. Brilliant. Thank you.
Great. Anybody else with any questions or follow-up here? I think, my mind always tries to go to what are the common threads across different conditions, different drivers, whether it's chromosome or monogenic, and just the fact that there could be some kind of a, not necessarily grand unifying theory, but something that kind of explains why you have to explain these fundamental features like more astrocytes, less neurons, and I know it's not possible to do the full dose response of all the chromosomes, but if like a little chromosome 21 can't, squeezing into the elevator causes this much problems, any chromosome that's larger, you just, I guess, is it stand to reason that if you had, or is there just, okay.
I mean, nature has already done this, right? So 3 chromosomes are technically make it to birth and people live. This is 21, 18, and 30. And so when you look at them by just genomic size, 13 is way too big, 21 is very small. But when you look at them on the amount of genes that they have, right, the genetic content, and if you were to rank every chromosome, 21 is the smallest, and then there's 18, and then there's 13. So the idea is that maybe I need to make this clear. We're not thinking that the whole disease is driven by this genomic imbalance drives everything, right? We're saying that every disorder has their own unique signature that is associated with the upregulation of genes that they have. But then there's also this common feature, which is probably associated with transcriptional, translational, replicative, and structural stress that's being introduced all together. So there are cell types that are very sensitive to changes in all of these features, which probably leads them to a very similar path. But then the unique gene expression makes each disorder sort of unique. Because even if you look at, let's say, Edwards, Patau, and Down syndrome, individuals have craniofacial, features that are unique, but each disorder has its own unique structural feature, right? The craniofacial as a whole is impacted by aneuploidy, but each one has its own unique. So the broad strokes are the same, right? Like the sensitivity is there. It's just we're trying to figure out, in addition to that unique feature, what's this underlying commonality?
So maybe one other final question about, from a drug repurposing point of view, what could be actionable from this right away? And, I asked this to Ari about like FGFR receptor inhibitors. And if you have overexpression of a target and you have a drug that inhibits that, you'd think that maybe that's a good therapeutic rationale. Is there anything from the senolytics or where you would say, what would make you think that this is something that could be actionable, in someone who's already born, and then even considering something intervening earlier? What's that level of evidence that you'd be looking for? Or is this not something, or you think that's kind of foolhardy to think you could attack this with repurposing?
I mean, to be honest with you, I just don't know. We don't have the data that's required for us to say this way or that way. I just do not know. I mean, I'm hopeful that we find something that would be, targetable and deliverable at any age time point, right? Like we're hoping that we can find therapeutic avenues for each group. It's just too early for us right now. But we're working on it, trying to expand beyond Down syndrome. And we have all of the aneuploidies now. So we have from chromosome 2 all the way to the sex chromosomes, triplicated, monosomies. We have these really crazy sort of genomic imbalances that are not viable, but we want to study the biology underneath them. We're utilizing copy number variations. This includes Pitt-Hopkins syndrome, DUP15Q. We have AP now, and so we're doing our best to get a handle on what's happening in neurodevelopment and brain function. We hope that we'll have something soon, at least to guide us of where the next steps would be.
Well, I really want to give you a round of applause again. This was a great, great session, and I think you also respect people who respect scientists who will say, I don't know, when you ask them a question, because then you trust them more when they do answer. And when you did kind of start to, hypothesize about, the MPC and the gain of function and the senescence, like you had very, very clearly thought this through. So I definitely respect that. And I hope that sets an example for our community here. So yeah, this was wonderful. Professor Meharena, thank you again for your time.
Thank you.
Yeah, thank you so much, Hiruy. We just, we're really excited to get everything started with your lab, and thanks for your patience. And I'm so glad to have AP caregivers at these meetings too. It's just shows how important all of the researchers are and how important we all are collectively as a community to just continue keeping an open space to communicate and exchange ideas.
Yeah, thank you. It's been wonderful to be part of this community. I'm glad.
And just to end with a few program notes here. So we'll finally be releasing our Project AP research plan. It will be going out this week. And so I look forward to seeing links there. And thank you to all the scientists and researchers here who helped review that. And then, I don't know, Whitney, do you want to preview who's up next for the next roundtables? Do you have that info handy here, or give people a sense of what's...
I believe it is. Our colleagues at the clinic in Colorado. So Scott Demarest and I'm sorry, I forget the other woman's name. Do you
Okay.
know?
So I can chime in. So the co-directors at our multidisciplinary coordinated care clinic in Colorado, Margarita Sines, who's a physician and geneticist there, and Dr. Scott Demarest, who's a neurologist and epileptologist, will be talking a little bit about clinical care and kind of thinking about how we can work together to get closer to some sort of standard of care guidelines and how, you know, natural history is really important. So I think that'll tie in nicely to what ultimately some of the researchers, including Hiruy, might need to see for phenotypic data and what's actually clinically observed in the 8P heroes over time. And then also we have a student MD PhD from Duke who's been helping with our actual registry and kind of trying to look at potential insights and areas of, you know, data, frankly, that, you know, might be gaps in knowledge that we still need to populate. So the three of them, oh, and her name is Mackenzie Beeman. So we're just happy to have her actually volunteering her time with the Chromosome 8P registry as well. So it'll be a nice conversation, slightly different from in the past. And so I hope that many of the families join that as well and just ask questions. And that's November 28th, correct, Whitney?
Yep.
Okay.
Great. So we look forward to seeing you there. In the meantime, we will share this video. We'd love to get more feedback and questions. So again, but I appreciate everyone who participated live, and thanks again to all the researchers who asked great questions. And when you're up in the hot seat next, then you'll get the same treatment and we'll get the same, I think, high level of conversation and creativity. So thanks everyone. Have a great rest of your day or evening, and we'll see you soon.
Bye, everyone.